Yes. Sei e sei. Buonasera a tutti e benvenuti al sesto seminario dell'edizione 2023 del ciclo di Orizzonti Medicina e Biologia. Sono Giulio Orlando e questo è il mio collega Luca Occhini, due studenti del secondo anno di Scienze Mediche, dell'Istituto di Scienze Mediche presso la Scuola Superiore Sant'Anna di Pisa. Prima di iniziare vorrei brevemente presentarvi questa nostra istituzione per quelli che tra voi non la conoscessero. La Scuola Superiore Sant'Anna è un'università pubblica alla quale gli studenti possono accedere esclusivamente per merito, attraverso un selettivo concorso nazionale. Gli studenti della scuola hanno l'opportunità di associare la loro carriera universitaria ad un percorso appositamente creato sulla base dei loro interessi. Ciò avviene all'interno di un ambiente multidisciplinare, altamente stimolante, dove gli studenti di diverse classi accademiche si ritrovano a condividere gli stessi corsi, gli stessi progetti e a vivere nello stesso ambiente. Hanno quindi la possibilità di confrontarsi, di crescere insieme sia sotto il profilo umano che sotto quello accademico, in un ambiente dove nuove idee nascono e maturano. Nonostante questa istituzione sia stata fondata solo 35 anni fa, in breve tempo ha raggiunto un alto prestigio nel panorama internazionale, sia in termini di educazione che di ricerca, essendo così menzionata nei principali ranking universitari. Ad esempio, nell'ultimo Times Higher Education World University Ranking, la Scuola Superiore Sant'Anna si conferma tra le prime università in Italia e nella top 15% globale.
La nostra è un'istituzione in costante cambiamento e offre la possibilità ai suoi studenti di rimanere al passo con le più innovative scoperte e di approcciarsi direttamente alla ricerca grazie a convenzioni con prestigiosi laboratori e importanti università nel mondo. La scuola inoltre si propone di offrire una formazione in ambito medico quanto più ampia possibile, intersecando il percorso medico con l'ingegneria, le biotecnologie, l'economia e l'etica, offrendo sempre nuove opportunità ai suoi allievi in un clima di interdisciplinarietà. Questo fervido ambiente culturale ha stimolato anche l'istituzione della serie di seminari intitolata appunto Orizzonti in Medicina e Biologia. Queste conferenze ci danno così l'opportunità di ospitare molti ricercatori brillanti e devoti alla scoperta medica che ci rendono partecipi della loro esperienza di ricerca nell'ambito biomedico e del loro percorso di vita. Colgo anche l'occasione per ricordare che questa è l'11ª edizione di questa iniziativa, la quale, grazie all'impegno e alla passione di tutti coloro che hanno contribuito in passato e continuano a farla nel presente, è cresciuta costantemente negli anni raggiungendo un pubblico sempre più vasto e poliedrico. Prima di iniziare l'incontro ci piacerebbe pertanto condividere con voi un breve filmato riassuntivo della scorsa edizione e di quelle passate.
Penso che Orizzonti sia un'occasione molto preziosa per noi allievi e in generale per tutti coloro che partecipano alla conferenza. Questo perché, come suggerisce anche il nome, è un'occasione per esplorare quelle che sono le frontiere della ricerca in ambito biomedico. L'ambito è anche molto ampio, gli ospiti, che sono sempre di grande spessore, trattano argomenti di tutti i generi e quindi è un bellissimo momento per ampliare e approfondire gli interessi di ciascuno.
Allora, fare il referente di Orizzonti è stata sicuramente una bellissima esperienza e anche molto arricchente. Prima di tutto perché imparare a organizzare e gestire una conferenza è una di quelle soft skill che ci potrà tornare utile in futuro nella professione che poi sceglieremo, e secondo poi perché permette di conoscere e parlare con queste persone che prima di tutto sono scienziati con la S maiuscola e secondo poi sono anche persone in grado di dare consigli che si vede derivano da un'esperienza di una vita ricca, piena e anche accademicamente perfetta.
Da Orizzonti mi aspetto di venire in contatto con persone che hanno a che fare quotidianamente con il mondo della ricerca e di apprenderne non solo i traguardi in ambito accademico ma anche l'esperienza di vita. Penso sia una grande opportunità che permette di mantenere il focus sulle sfide attuali della medicina, sentendole presentate da chi ci lavora ogni giorno.
Ringraziando il professor Zucker per aver accettato il nostro invito, lascio la parola al professor Vincenzo Leonetti che si occupa di questo progetto e che introdurrà appunto l'ospite. Vi ringrazio per l'attenzione e vi auguro una buona serata.
Buonasera a tutti. Welcome to everybody. And today is a great honor for me to introduce Professor Irving Zucker. Thank you to be here. Professor Irving Zucker is one of the great physiologists that we have actually. And he tried to investigate in different fields using a multimodal approach, even animal model. And he tried to target different areas of specialization like heart failure, hypertension, renal function, neural control of the circulation, and oxidative stress. But to reach this great scenario, you need to start when he was a student. And like you are a student now. And he received his bachelor in 1965 at the City College of New York and in biology. And then the master degree in 1967 at the University of Missouri at Kansas City, always in biology. And at least in 1972, he received his PhD degree at New York Medical College in New York, a PhD in physiology. So this was his education. But he had several experiences pre and postdoctoral. During his pre-postdoctoral training, he was from 1968 to 1972 NSF predoctoral trainee at the Department of Physiology of New York Medical College. And for 1 year, he was NIH postdoctoral fellow at Department of Physiology and Biophysics at the University of Nebraska Medical Center. So it's like the students here. So he moved from a student to education and tried to grow. He had several academic appointments. Actually, he's a professor at the Department of Anesthesiology of the University of Nebraska. But the first appointment was as an assistant professor at the Department of Physiology and Biophysics at the University of Nebraska Medical Center, Omaha, Nebraska. We were around 1973. That is my year of birth. So 1973 is a long time ago. And then he was associate professor from 1980 to 1983, as assistant professor at Department of Physiology at the same institution. And then he was, and he actually, maybe recently not, but from 1989 to 2019, he was chairman at Department of Cellular and Integrative Physiology at the University of Nebraska. He received several honors because he made several discoveries in the field of cardiovascular physiology, even studying the relationship between the heart and the brain, so the brain-heart axis. And several prestigious recognition from the American Heart Association, where he was chairman of the research committee. Far away, he started pretty soon in 1979. And actually, and he went on in his career, even in other prestigious society like the Heart Failure Society of the American, where he received an award for outstanding contribution to the College of Medicine. Even the Marcus Award Selection Committee from the Council of Basic Cardiovascular Science of the American Heart Association. He is fellow of the American Heart Association, fellow of the American Physiological Society. In 2003, he was president of the Association Chair of the Department of Physiology. And he was also editor-in-chief of the prestigious American Journal of Physiology, Heart and Circulatory Physiology. He is pretty active as a reviewer, actually, for several prestigious journals, and he is member of several organizations, like the National Institute of Health, first of all, for several sections, even trying to evaluate grant application. And today, he will be here as a professor for one day at Scuola Superiore Sant'Anna, and they will talk about his career and the discovery that he did and how the role of physiology changed over time. Thank you.
Grazie per questa gentile introduzione. Parlerò inglese, however. I'm really grateful to have the opportunity to visit you. I want to thank both Professor Leonetti and Professor Recchia for their kind invitation. And I hope I can convince you that understanding cardiovascular physiology in the setting of disease is multimodal. And I've taken a historical approach to this because I know there are varied people in the audience that are at all levels. So I'm going to start off with somewhat of a historical approach. Let me see if I can advance the slides here. By the way, if anybody needs to contact me, this is my email address. And I will leave these slides with Professor Recchia in case anybody wants to review them. So this is not advancing slides. Okay. So I want to start off with a bit of a historical overview. I mean, now that I'm here talking in Italy with this great, rich history of anatomy and physiology, we have to go back and think about how the cardiovascular system was organized in the days of Galen, right? It's 129 to 216 AD. And the old story was that the liver was responsible for making blood and circulating it to the various tissues, and the lungs provided some sort of humoral substances. And then there were these mysterious pores between the septum of the right and left ventricle that allowed these substances to go to other parts of the body. And this was a concept that was not based on experimentation, but was based on an idea that Galen put forward many, many years. And it was the standing idea for hundreds and hundreds of years. But then, But then during the Renaissance, there was much more focus on human anatomy. And it was actually Leonardo da Vinci and Andreas Vesalius who did detailed anatomical descriptions of the human body. But it's interesting that even though Da Vinci never saw these pores in the ventricle. He acknowledged them because everyone was afraid to go against the Galenic theory of how the blood circulates. If you go back and you look at Leonardo's drawings in his notebooks, they are incredibly detailed. All the coronary arteries, all the valves, but no mention of pores in the septum. It wasn't until William Harvey in 1628 who wrote De Motu Cordis, in which experimentally he showed that the circulation was circular, right? And that there had to be a way of going from the lungs back to the heart to the systemic circulation. Harvey didn't know much about the microcirculation or about the capillary structure. And of course, he described valves in the veins, and these are classical kinds of experiments that many of you know. Now, as Professor Leonetti mentioned, I was editor of the American Journal of Physiology for 7 years, Heart and Circulatory Physiology. And during that time, I had the opportunity of having all of my associate editors visit our institution in Omaha, Nebraska for strategic planning meetings and to give symposia. And I wanted just to point out to you that both times, there's this guy in the middle of both meetings, Professor Recchia, who was the center of discussion. So the talk will focus on several areas, and I apologize in advance if it's a little bit long. I'm going to give you an overview for the students of the concept of heart failure, how to identify heart failure, a little bit about reflex control of the circulation, oxidative stress, some more recent work that we've been working on a transcription factor called NRF2 and its role in mediating changes in oxidative stress, both in the brain and in the peripheral circulation, and the role of microRNAs and exosomal regulation as organ crosstalk between the heart, the injured heart, and the brain. And then finally, a few things, if we have time, on exercise and skeletal muscle. Okay. So you can define heart failure in many different ways, and this is just a variety of different definitions. Early on in 1933, Thomas Lewis said it's a condition in which the heart fails to discharge its contents adequately, and of course that's correct. Paul Wood in 1950 said it's a state in which the heart fails to maintain an adequate circulation for the needs of the body, despite an adequate or higher filling pressure. Right? And finally, finally, the American College of Cardiology defines heart failure as a complex clinical syndrome that results from any structural or functional impairment of ventricular filling or ejection of blood, right? And that's an important distinction today because we, as I'll show you in a minute, we define heart failure in several different ways depending upon symptomatology. So there are two types of heart failure. There is heart failure with reduced ejection fraction, and we define ejection fraction echocardiographically as the ratio of the stroke volume minus the end diastolic volume, that is the volume in the heart at end diastole, times 100. And it's expressed as a percentage, and everybody knows that normal human beings have a ejection fraction of about 50 or 55%, maybe sometimes 60%. But now we have focused in more recent years at another entity called HFpEF, or heart failure with preserved ejection fraction. So these patients have normal ejection fraction, which is about 50%. They show evidence of abnormal diastolic function. That is, relaxation time is longer, and they present with symptoms of pulmonary congestion. Okay. This is an entity that is very different from what you would get with a myocardial infarction, perhaps, or with structural kinds of heart failure or myocardial hypertrophy that's gone too far. It turns out that 50% of patients with symptomatology can be diagnosed with HFpEF or heart failure with preserved ejection fraction. In the United States, heart failure is a huge issue. About 6 million patients in the United States are living with heart failure, and the yearly incidence is about 600,000 new patients per year with heart failure. And of course, there's good medical therapy for heart failure, and with end-stage heart failure, there's also therapy, which is cardiac transplantation and LVADs and things like that. Economically, it's a huge burden, and it's expected to increase dramatically over the next 6 or 7 years. So it's something that we need to understand more about. We need to develop new therapies for. And this just shows survival data for different stages of heart failure. And you can see in the most severe stage, there's only about a 20% survival rate. And end-stage heart failure has a 1-year mortality equivalent to pancreatic cancer, which is one of the more virulent types of cancer. Heart failure comes in all forms. There's ischemic cardiomyopathy that I think most of us are familiar with, the most common. And there's non-ischemic, which is cardiomyopathies from either acquired reasons or genetic reasons. And there are many different reasons. There's some heart failure that's reversible, like peripartum heart failure. There's cardiac sarcoidosis, the laying down of sarcoid protein and things like that. And there's toxicity. There's diabetic cardiomyopathy. There's alcohol-related cardiomyopathy. So it is an important phenomenon. And one of the things that occurs when cardiac output falls is that there is an immediate compensatory reaction to that decrease in cardiac output, which is activation of the neurohumeral axis in the heart. So remember, everything is negative feedback. Everything in physiology is homeostatic, right? Positive feedback doesn't work. I'll show you an example of positive feedback. It's mostly pathological. So the body tries to maintain cardiac output by activation of the neurohumeral system, which includes the sympathetic nervous system, the renin-angiotensin system, and several others. And this is beneficial in the short term. But if it's prolonged, it's counterproductive. And a negative feedback system turns into a positive feedback system. And it's a vicious downward spiral in which cardiac function worsens and worsens and worsens. And in fact, modern therapy for heart failure is to target the neurohumeral axis, which means beta blockers, the renin-angiotensin system, the vasopressin system, et cetera. Okay. So when we think of heart failure, we have to think about everything that contributes to the maintenance of this arm of blood pressure control. Remember, blood pressure is regulated by cardiac output and peripheral resistance. And each one of these have ways of being regulated. For cardiac output, of course, it's stroke volume and heart rate, and contractility is something that can be regulated by intrinsic muscle proteins as well as catecholamines and the sympathetic nervous system. We have to think about venous return and cardiac filling, and that goes back to blood volume regulation, venous turn, and the whole idea of the renal axis. So when one tries to work out mechanisms, we have to think about this, and I'm sure all of the students in the room are already familiar with the Frank-Starling curve and the relationship between a cardiac filling or left ventricular end diastolic pressure and stroke volume or cardiac output. And when the heart fails, we don't just move up and down this curve, we move to a flatter curve. And these are things that have worked out in the 50s and 60s, and this is really the basis for understanding how to treat patients with heart failure. What you want to do is you don't want to treat these patients by moving them up and down. You want to bring them up to a better contractile curve. Okay? And if you were to take this to the other extreme, during exercise training, you'd move this curve to the left, or during not exercise training, but acute exercise. Okay? So that kind of ends the little tutorial, and now I want to step back 50 years and tell you how I developed my own career with the idea that understanding the regulation of the peripheral circulatory system will help us understand heart failure and the treatment of heart failure. So when I was a graduate student, I worked with this gentleman whose name is Gabor Cayley. Dr. Ecker knows him very well. And his interest was the microcirculation, the renin-angiotensin system. And we decided to do some experiments to answer the question whether there was a sensor of sodium located in the central nervous system. Was there, we knew about osmoreceptors from 1947. We knew that the brain, especially the hypothalamus, could interpret changes in osmolality. But heart failure is associated with abnormal sodium balance. The question was, is there a sodium sensor? So what we did is we prepared dogs with cannula in the third ventricle, which when injected into, will change the environment around the hypothalamus, especially the paraventricular nucleus. And one experiment we did was we looked at sodium excretion. The question was, does this sodium sensor in the brain regulate sodium excretion, which in turn would regulate blood volume? And what we found is when we gave hypertonic saline into the third cerebral ventricle, the animals responded with an increase in sodium excretion. And that lasted for about 30 minutes, and that sodium excretion returned to baseline in about an hour. If we cut the nerves to the kidney, if we denervated the kidney, we abolished this effect, suggesting that there was a neural connection between the central nervous system and the ability of the kidney to respond to catecholamines to regulate sodium excretion in the tubular system. So that was kind of this is my PhD thesis. This is my first introduction into cardiovascular medicine. And then I got to thinking, how does the body regulate volume? How does the brain know what our plasma volume is at any moment in time? So there was a group of investigators in our department in New York Medical College, John and Hazel Coleridge, and they were interested in sensory function of the cardiovascular system. And I learned how to record electrophysiological activity from the vagus nerve by making small slips of fibers, putting them on electrodes, and recording action potentials. It turns out there are receptors lining the atria and the ventricles, but mostly in the highly distensible atria that are mechanoreceptors. And here's what they look like. There are modulated, sometimes non-modulated axons, and this arborization of sensory endings. Now, when you think that these are embedded in a structure that's highly distensible, when you stretch that structure, you mechanically depolarize these endings, which sends action potentials up the vagus to the central nervous system. So the brain not only knows about blood pressure, oxygen, it knows about the distension of the heart by virtue of this highly distensible mechanoreceptor system. So I came to Nebraska, and along with this fellow, Joe Gilmore, who worked at the NIH for many years with Stanley Sarnoff, defining cardiac function, we decided to look at how these atrial receptors behaved when we altered myocardial contractility. And this was done in both an acute way and a chronic way. So the first thing we did is we recorded neural activity from the vagus nerve, and here are action potentials. And we expanded plasma volume. And if you look at left atrial pressure here, you can see as we increase plasma volume, there's an increase in left atrial pressure, not very much, but an increase. And if you look at discharge sensitivity, for each cardiac cycle, there's an increase, and you see it becomes quite continuous after a while. Okay. If we do another experiment in the bottom panel, where we ligate a coronary artery and we depress myocardial function, you can see this right here. This is left atrial force, and here's this is left atrial pressure. You can see that left atrial pressure, mean pressure goes up. You can see that the discharge also increases. So when one plots this data as the relationship between left atrial pressure and discharge, whether you do coronary occlusion or volume expansion, that falls along the same curve for a given receptor. These are two different receptors, and you can see they overlap. So this suggests that it is distension, whether you do it by volume or by depressing contractility, that is responsible for this discharge. And in fact, in Joe Gilmore's lab, we were able to reduce atrial contractility by giving large doses of propranolol. I mean, very large doses of the beta blocker propranolol. And you can see, if you look at here at right atrial force, and these were something called strain gauges, which are put on the myocardium, and you can keep the length constant, so you can measure the force that independent of the length. You can see when you depress, in this particular, there was one spike per cardiac cycle that happens before the QRS, right after the P wave, in concert with atrial contraction. But when you depress atrial contractility, atrial pressure increases, and you see now this one spike is 4 or 5 spikes per second. And once again, if you plot the change in left atrial pressure with the change in discharge, it falls along a continuum which is coincident with atrial size. So this is clearly a mechanoreceptor effect. Now, we then say, well, what happens to these receptors if you make heart failure and you reverse heart failure? Can you make them normal again? And we did this experiment in which, and this is primarily an anatomical experiment done in animals, where we opened an AV fistula between the aorta and the vena cava. So we had this great big heart, huge heart, high output failure, lots of venous congestion, lots of pulmonary congestion. And when we look with the fistula open, you can see that these receptors are completely abnormal. The axons are there, but the endings are pulled apart. Then when we close the fistula, because we can do this with a Dacron graft, close the fistula, you can see that over time, the left ventricular pressure comes down, the cardiac output comes down, and these receptors now look normal again. Okay, so this is a reversible phenomenon. The last experiment we did in this now is 1979, we showed that if you inflate a balloon in the left atrium of a normal animal, you get a diuresis. And this is because arginine vasopressin is decreased. But in a heart failure animal, when you inflate the balloon, there's no diuresis. Probably because vasopressin or antidiuretic hormone is very high in heart failure. And even though you can get it depressed, it's at such a high level that you really cannot see, a diuresis, right? And these are the plasma osmolalities down here, okay? So that was a whole era of experimentation. Then we got thinking about the autonomic nervous system some more. And we said to ourselves, okay, for those of you for the students that may not be familiar with the organization of the autonomic nervous system, remember there's the sympathetic nervous system, which has short preganglionic and long postganglionic neurons. And then there's the parasympathetic nervous system, which has long preganglionic and short postganglionic neurons, right? And sympathetics, at least for our discussion here, they innervate lots of tissues, but of course, for our discussion, the heart, the SA node, and myocytes is the most important, right? And the parasympathetic nervous system will activate or slow the heart by its innervation of the SA node and the AV node. But there are many other effects of vagal stimulation, which is all cholinergically mediated. So it was shown years and years ago by Jay Cohn and David Ferguson at the University of Iowa that if you look at patients that have been diagnosed with heart failure, compare their level of plasma norepinephrine to normal patients, you can see that there's a relationship between plasma norepinephrine and survival. Those patients with the highest level of plasma norepinephrine have a very poor survival profile. So this is like 2 years after diagnosis of heart failure compared to patients that have lower levels of norepinephrine. And if you use microneurography, which in these days was somewhat in its infancy, you can show that as cardiac function worsens, the bursting activity of sympathetic nerves, in this case in muscle-targeted nerves in the perineal nerve, increases. So we could show the same thing in our animal models. Now in animals, we didn't do microneurography, but we can measure norepinephrine. We could measure sympathetic nerve activity electrically, mostly going to the kidney, but sometimes to the heart. And we asked these questions about the arterial baroreflex, the chemoreflex, so these are inhibitory reflexes, excitatory reflexes, and we were especially interested in this cardiac sympathetic afferent reflex, in which endings enter the spinal cord and are excitatory to sympathetic outflow to most of the peripheral circulation. These are kind of nested reflexes that have their pathways in both the spinal cord, of course, in the efferents to the heart and the stellate ganglion and some cardiac nerves. There are monosynaptic reflexes through the dorsal root ganglia into the spinal cord, and then there are projections up into the brainstem and higher centers that regulate sympathetic outflow under a variety of conditions, including emotional stress, exercise, and what have you. Now, if you go back to William Harvey, he and King Charles, they had a patient that had part of their heart, this patient had part of its heart exteriorized, and they could touch it. And they said, look, we can touch this patient's heart, and he doesn't feel anything. So therefore, the heart must have no pain sensation. And he wrote that he concluded that the heart itself appears to be insensitive. Well, certainly we know that's not true, right? Certainly during coronary ischemia, you have angina, you have chest pain, and that's a well-known phenomenon. And oftentimes that's how patients become aware of their disease. The afferents that sense cardiac pain are located in multiple layers, mostly in the superficial layers of the myocardium. They enter through the dorsal root ganglia. They transect the spinal cord and go up the spinal thalamic tract, the spinal retricular tract, give off projections to the brainstem, and make it all the way up through the thalamus to the cortex, where you get the sensation of pain. Now, there are many things that stimulate these afferents, and here's just a partial list of the kind of things. And it turns out these are all things that are released during coronary ischemia, right? Including hydrogen ions, oxygen radicals, potassium, etc. So we became very interested in this phenomena. So together with a postdoc, Wei Wang, in the mid-90s, we showed that in animals with heart failure, if you apply bradykinin onto the heart, which is an analgesic agent that activates these nerve endings, you find that you get an increase in sympathetic nerve activity, which is much more pronounced in animals with heart failure. And here you can see the comparison of 2 different doses of bradykinin on renal sympathetic nerve activity. And you can see even in lower doses, there's a bigger effect in animals with heart failure. So that took us down a long number of studies and projects. And this just shows that you can also see the same effect with capsaicin, which is a derivative of hot pepper, and you can see that if you put capsaicin onto the surface of the heart, you get much larger effects in heart failure. And furthermore, if you simply put lidocaine onto the surface of the heart, you can inhibit sympathetic nerve activity in animals with heart failure, but not very much in normal animals. So that suggests that these afferents are sensitized in the setting of chronic heart failure. Now, this may be hard to see, but this is an image in a movie of the dorsal root ganglion. This happens to be a lumbar dorsal root ganglion, but it's similar in the thoracic segments. And the yellow and red neurons are expressing a protein called TRPV1, or transient receptor vanilloid potential 1. TRPV1 is a nonspecific cation channel that is highly permeable for calcium. Okay? And most people believe that it transmits sensations of heat, burning. So people with, for instance, with claudication from peripheral arterial disease will have pain upon walking, and that is mediated to a large extent by activation of the TRPV1 receptor. Now, why am I showing you this? Is because if you give an agonist of the TRPV1 receptor, like capsaicin, or another agonist called RTX, which stands for resinaviratoxin, which is 1,000 times more potent than capsaicin. Now, if you look at fluorescence, and this is calcium fluorescence, you can see that at 1 micromolar, you get a nice peak in calcium fluorescence. You can see this here. For RTX, at 1.6 nanomolar, you get this enormous influx of calcium into these neurons, which is sustained. If you actually look at the calcium current, you can see that it is sustained for quite a while. It's very slow to turn off. So we said, if we use this substance, maybe we can destroy these neurons and take away this cardiac afferent reflex, and perhaps that would be beneficial. So what we did is we gave RTX to the surface of the heart, which opened up the TRPV channels caused calcium influx, which was toxic to the cell, right? We're not so much interested in TRPV1 as destroying these neurons. So using RTX, we can do this. And these experiments were done with a postdoc who's now a faculty member, Hanjun Wang, in the lab. And this experiment was very simplistic. We made animals with coronary ligation, myocardial infarction. These were done in rats, or they were sham. And then at the time the chest was open, we painted the surface of the heart with either RTX or vehicle. We let the animals recover. We did echoes periodically. We collected urine for norepinephrine measurements. And then we did terminal experiments at between 9 and 11 weeks later. Okay? And this just shows you, and I don't want to go through this table in time, but I just want to point out the left ventricular end diastolic pressure. So in the sham animals, it's about 5. You give RTX, doesn't change. In heart failure animals, the end diastolic pressure is about 20. But in the group that received RTX, it was much lower, right? It's almost within the normal range, right? So we took these animals and we did pressure volume loop analysis, which we put in a pressure volume catheter, and we could measure both systolic and diastolic function at the same time. And this end systolic pressure volume relationship defines the inotropic state of the heart. And what you can see here in sham animals treated with RTX, there's not much difference. But in animals with heart failure, you can see that if you compare the slope of the end diastolic pressure volume relationship to the sham, you can see it's much steeper, which indicates a stiffer heart, poor diastolic function. In addition, systolic function is impaired, right? However, after RTX, not much happened to systolic function, but diastolic function was greatly improved. And you can see that in the mean data here. End systolic pressure volume relationship, not much, but end diastolic pressure volume relationship quite restored. Okay, and you can see a similar thing in negative DPDT. Okay, so diastolic function can be determined by a variety of things, including calcium reuptake after contraction, including the degree of extracellular matrix, that is the degree of fibrosis. So we tried to look at that. This may be a little bit hard to see, but you can see that in animals with heart failure, this is what we call picroserous red staining. There's more fibrosis. And in the RTX animals, that fibrosis is less. This is a Masson's trichrome stain. You can see the degree of fibrosis in the myocardial infarction area. And what this shows is that after RTX, the degree of fibrosis in the intraventricular septum, as well as in the remote left ventricle, is reduced in animals with heart failure. And some of the proteins that are responsible for production of extracellular matrix, alpha smooth muscle actin, is also reduced by RTX. Fibronectin is reduced. These are Western blots, by the way. And TGF beta is also reduced, which is part of the extracellular matrix pathway. So, so this was very interesting to us because it pointed that there may be a way of reducing diastolic dysfunction. Now, please remember that this was not a therapeutic intervention in that we gave the RTX at the time we made the info, right? We have yet to do a therapeutic intervention, which is a much more difficult thing to do. Then we said, well, these afferent endings, they're loaded with neuropeptides. They secrete neuropeptides. When they're activated, those neuropeptides should be released. And many of these neuropeptides are vasodilator, change underlying capillary permeability, and cause plasma to move out of the capillary into the interstitial space. This plasma extravasation is a message for inflammatory mediators to move into the area. This inflammation participates in the extracellular matrix, and these neuropeptides are substance P and CGRP and several others. So then we said, okay, what happens if we give RTX to the ability of plasma to come out? And what we found is this may be hard to see, but if you put a small piece of filter paper soaked in bradykinin onto the surface of the heart in a sham animals, you soak in either bradykinin or capsaicin, and then you inject Evans blue into the animal. That Evans blue binds to albumin and comes out into the interstitium, because you can wash out all the plasma. And you can see if you can't see this, this is blue, there's dotted lines. If you do a myocardial infarction, you see that this whole area here is stained with blue, right? Either every 1 day post-MI or 1 week post-MI. But in animals treated with RTX, there's no blue in either the bradykinin capsaicin experiment or the myocardial infarction. Okay, and you can block this with an NK1 antagonist called SpanTi2, which blocks the effects of substance P. The other thing we found is that inflammatory mediators like IL-1 beta are reduced in animals treated with RTX, as TNF alpha also. Then when you look more closely at the extracellular matrix, in scanning electron microscopy, you can see that in heart failure, there is a huge increase in extracellular matrix, which is kind of reversed. In the RTX treated animals, matrix metalloproteinase 9 and 2 are upregulated, as shown here with zymography, and that is reversed with RTX, similar for MMP2, and the mean data are shown here. You don't see this at all in the sham group. And then finally, the important thing is that what happens to survival? So if we let these animals go for 30 weeks, we find those animals treated with RTX have a much better survival profile. The vehicle-treated animals start dying off here at 10 weeks, and as we go, we lose more and more animals. So about 50% of them are dead at the 27, 28, 30 weeks, whereas you can see that 90% of them were still alive at that time period. So to summarize this part of the talk, we think that one can intervene in this neurohumoral feedback system, remove some of these sensory endings in the myocardium, and improve cardiac function and reduce this positive feedback that leads to myocardial cell death and further declines in cardiac function. Okay. I want to switch gears just a little bit. And if anybody has any questions, we can wait till later, or you can stop me anytime. I want to tell you about some more recent work we've done and what we're continuing to do with reactive oxygen species, or reactive oxygen stress. You know, oxygen-free radicals can be made by a variety of sources. The two most important sources in the body is from the plasma membrane here through the enzyme NADPH oxidase. NADPH oxidase is a variety of proteins, P67, P47, et cetera, and and in this case, NOx2 or GP91. And what essentially happens here is that oxygen takes on an electron to form a free radical. That free radical can be released either into the extracellular space or into the cytosol. Now, the problem with free radicals is they are so highly reactive that they bind to proteins and to DNA and to lipids. The other source of free radicals is in the mitochondria. And as one transfers electrons from complex 1, complex 3, complex 4, free radicals are made in the form of superoxide anion. They are released both into the cytosol and accumulate in the mitochondrial space. Now, under normal circumstances, this is not a problem, right? The increase in metabolism of any tissue will bring about increase in oxygen free radicals. But we have ways of dealing with this. And the primary way is through the elaboration of antioxidant proteins and small molecules. So one of the questions we ask in terms of the regulation of sympathetic nerve activity is there more oxidative stress in the brain of animals with heart failure? And in collaboration with Li Gao, who I still work with, back in 2004, we used two different techniques to look at oxidative stress in an area of the brain called the rostral ventrolateral medulla. And the reason we chose that area is because that's where most of the presympathetic neurons are located that send projections down to the intermediolateral cell columns of the spinal cord. So we use this lucigenin assay, which relies on the production of a fluorescent product of lucigenin and dihydroethidium, which binds to DNA in the presence of reactive oxygen stress. And this was in a rabbit model of pacing-induced heart failure. And as you can see, the amount of free radicals was much greater in the heart failure animals, for both assays compared to sham animals. If we looked at the antioxidant enzyme mitochondrial superoxide dismutase in punches of the RVLM, or the cytosolic enzyme copper zinc superoxide dismutase, we found that in heart failure, there was a reduction in both of these proteins compared to normal. Disregard the exercise training, that's a whole nother story. And these are, again, Western blots. So then we had the idea, well, what happens if we record sympathetic nervous system? And one of the reasons we chose the rabbit is because they are really easy animals to record sympathetic nerve activity from in the conscious state, right? So these are all awake experiments. So the animal's standing there, we have electrodes coming out, we measure the sympathetic nerve activity. And you see that if we overexpress superoxide dismutase using an adenovirus injected into the RVLM, in a sham animal, there's no change in sympathetic nerve activity because it's already low. But in animals with heart failure that have augmented levels of sympathetic nerve activity, we get a dose-dependent reduction in sympathetic nerve activity. So that means that scavenging oxidative stress is a good thing. So everything in life is about balance, right? And the balance between antioxidants, antioxidant enzymes, and the production is what really we need to focus on. If we're under an assault of pro-oxidant stimuli, how can we manage that? And we came up with this idea 10 years ago or so, and and I won't go into the whole reasoning behind how this came to my mind, but there is a transcription factor called NRF2. It stands for nuclear factor erythroid-derived 2 like 2. We just call it NRF2. You'll also see it as NFE2L2. Let's just call it NRF2. It's located in the cytosol in conjunction with another protein called KEAP1, which is part of the E3 ubiquitin ligase system. So it's responsible for degradation of proteins. So under normal circumstances, NRF2 is ubiquitinated in conjunction with another protein called culin-3. It is that ubiquitination dissociates targets NRF2 and KEAP1 for incorporation into the proteasome, which degrades these proteins. So if there's no oxidative stress, you would expect NRF2 to be at relatively low levels, right? Because there's no immediate reason we need more antioxidant proteins. However, in the face of oxidant stress, NRF2 dissociates from KEAP1. And how does it do this? If you look at the structure of KEAP1, there are several cysteine residues that have sulfhydryl groups that stick out into the cytosol. Now, as you know, sulfhydryl groups can be oxidized very easily. And once those, and there are 3 specific ones, once they're oxidized, there's a conformational change in the molecule. And that causes the release of NRF2 from the KEAP1 complex. So KEAP1 is an inhibitor, right? Once NRF2 is released, and some activators that are known, some drugs can do the same thing, once we have oxidative stress, NRF2 is phosphorylated, KEAP1 then becomes degraded, NRF2 moves into the nucleus, which binds to the antioxidant response elements of at least 200 genes, probably now up to 500 genes. Some of them are anti-inflammatory, some of them are antioxidant. Okay? There's a whole other story about the relationship between NRF2 and NF-kappa B, which is part of the anti-inflammatory cascade. So the bottom line is that activation of NRF2 is a good thing if you have high levels of oxidative stress, because it upregulates things like heme oxygenase 1, NQO1, catalase, and SOD. One of the things that 2 students, a postdoc and a student in the lab, found was that they looked in the brain, in the rostroventral medulla, and they looked at animals with heart failure, both in the rat and in the mouse. They discovered a relationship between the ejection fraction, or the severity of disease, and the level of NRF2 expression. And animals with the poorest ejection fractions had the lowest level of NRF2 expression. So that right away excited us, because we thought, well, maybe we could intervene and raise the level of NRF2 as a therapeutic intervention. So the first question we asked was, if you knock down NRF2, in the RVLM specifically, will it change oxidative stress, and will it change sympathetic nerve activity in normal animals? So we did this experiment. We got NRF2 floxed animals, and I apologize if you don't know what a floxed animal is, but it's a technique in which we can delete or overexpress a specific protein in conjunction with a protein called Cre recombinase. And we obtained this line from Siam Biswal at Johns Hopkins University, and in conjunction with Li Gao, what we did is we took these NRF2 floxed mice and we injected a virus that codes for Cre recombinase, in which there was a GFP tag. And we injected that directly into the rostroventral medulla. Okay, so we were able to knock out NRF2. And this is just some validation data showing when we look at we take punches of the RVLM in these mice, and we look at the message It's reduced in those animals receiving Cree. The protein, by Western analysis, is reduced. This is the area we targeted, the RV11. It's a tiny, tiny area. And this, I know it's hard under this lighting to see, but what I want to show you is that the GFP is in the rostroventral lateral medulla, and NRF2, which is in the red stain, is not shown. Okay, and then if we look at oxidative stress using electroparamagnetic spin resonance, we find that when we knock out NRF2, there's an increase in the signal. This is a very noisy signal compared to other EPR spectra you may have seen, because the amount of tissue we have to work with is so very small. So then we telemetry these animals. We put in radiotelemetry transducers, these are awake animals. We recorded their blood pressure 24 hours a day, right? And this is the blood pressure profile. And you can see they have a mean arterial pressure of about 95 or 100 millimeters of mercury. This is the daytime. This is the nighttime pressure. In the animals where we knocked out NRF2, they became hypertensive with very little change in heart rate. We did not expect this at all. This was a big surprise. So one thing it says is that even though without oxidative stress, there are low levels of NRF2, if you take that away, whatever the level is, it releases sympathetic nerve activity, and you get an increase in blood pressure, both daytime and nighttime. And in fact, we also showed that if we collect urine and measure their norepinephrine excretion, that there is an increase in sympathetic nerve activity. And we went further, we recorded sympathetic nerve activity. There's only a few people in the world that can do this in mice, and Li Gao is one of them. And you can see what happens, that in animals with heart failure, there's very high sympathetic nerve activity. You can see that here. And when you give, I'm sorry, not with heart failure. In animals in which the in NRF2 is knocked out, sympathetic nerve activity increases markedly, and the baroreflex function is markedly impaired. Remember, when you raise blood pressure, sympathetic nerve activity turns off, and you cannot see this if you delete NRF2. So the next question was, what if you do this in heart failure animals? The opposite side of the coin. What if you raise NRF2 levels? So the approach we took here was to knock down KEAP1. So if you knock down KEAP1, you raise NRF2. And I know it's getting a little bit long on time, so I'm going to try to go through this rapidly. But the bottom line here is that you can see that norepinephrine is reduced, sympathetic nerve activity is reduced if we knock out KEAP1. So it happens in normal animals when we take away NRF2, and in heart failure animals when we take away KEAP1. So this is kind of a summary diagram showing that NRF2 can modulate sympathetic nerve activity in animals with and without heart failure. Okay, last part of the story is I had another assistant professor that I collaborated with, Shanghai Tian, who said to me, how does the signal get from an injured heart to the brain, and can you modulate NRF2 activity in the brain? Why does NRF2 go down, right? If anything, with high levels of oxidative stress, we should have more NRF2. But yet when we look at the protein, it goes down. So he came up with this idea that there is some signaling that's not neuronal that transfers material from the heart to the brain. And he had previously shown that within the heart, there's in the injured heart, there is extracellular vesicles that are released that contain all sorts of cargo that can communicate between different types of cells. And he came up with the idea that the myocardium, when it's injured, sorry, let me go back. The myocardium, when it's injured, releases these extracellular vesicles that get to the brainstem and alter sympathetic nerve activity by reducing nerve 2. So he took a look at this, and he looked at the database to see what microRNAs. Well, let me show you this first. Here's a rat without heart failure and with heart failure. And if you look at nerve 2 in the at nerve 2 levels in the messenger RNA, we find that the messenger RNA is high, but when you look at the protein, it's low. So how could that happen? The only way it can happen, or one of the ways it could happen, is that there are microRNAs that inhibit the translation of the message to the protein, as you can see here. And when we looked at antioxidant enzymes, we find that they were all decreased. So the message is one thing, but it's the protein that's really important. And I know you can't see this, but these are indices of oxidative stress. So one of the things he looked at in the rat hearts is he did a database search, and he found that there were specific microRNAs, 27A, 28-3P, and 34A, that have NRF2 as a target. And we found that in these animals with heart failure, there were increases in the hearts of these microRNAs in the non-infarcted part. There were other microRNAs that have NRF2 as a target which were not changed. Furthermore, we were able, in collaboration with our cardiovascular biobank, get human heart tissue. These are patients that had LVADs implanted, so the cores were frozen. And here we have human heart tissues in which we measured these same microRNAs, and compared to donor hearts, which we don't have too many of, you can see that these same microRNAs were increased both in ischemic cardiomyopathy and dilated cardiomyopathy. But in humans, there were additional microRNAs that were elevated, like 142, 144, and 153. So all of these have in common that they target, at least one of the proteins they target is NRF2. Okay, and NRF2 targeted microRNAs were also shown to increase in the brain. So this shows you in animals with heart failure, there were increases in these same microRNAs. We assumed that they got there by transport either through blood or through extracellular vesicles. So we looked at the extracellular vesicles. This is an electron micrograph of what they look like. The average size distribution in this case was between 80 and 100 nanometers. They express all of the markers of extracellular vesicles. And we found that they're increased, that the cargo from these extracellular vesicles have the same increase in heart failure of these microRNAs. In addition, we found that there are cardiac-specific microRNAs, like microRNA 1, 499, and 208, that are contained in the EV cargo. And this just shows that similar, if we look in both humans and animals, we see large increases in plasma levels of these same microRNAs in the setting of heart failure. And here you see in humans the same sort of thing. So this seems to be a ubiquitous thing. Now, the last thing I want to show you is the ability to demonstrate that if you give extracellular vesicles and you label them with a dye and then you image them, they get into the brain. So you can see if we give an IV, if we've done this IV, IP, intranasal, you can see that the brain expresses fluorescence to this PKH26 label. And then if we look specifically at the RBLM, I know this is hard to see, you can see little red dots here, which are in the neurons. They're not just on the surface of the brain, they're taken up in the neurons, and you can see this in high power here. We can also show this using a red-green reporter mouse, in which we can show that only particles coming from the heart tied to the myosin heavy chain promoter winds up in the brain. So there's no question that EVs from the brain can enter the heart. And then the final experiment we did is that we injected EVs from heart failure animals into the RVLM of normal animals and showed that we get an increase in norepinephrine excretion. But if we block these microRNAs with antigomeres that target those microRNAs, not only do we inhibit norepinephrine production, but we also inhibit oxidative stress. And then finally, same experiment, we can reduce, we can reduce sympathetic outflow with these antigomeres. And I won't bore you with all the rest. So that kind of sums up a lifetime of work. This is the kind of the gestalt we're working on right now in terms of what we think happens. The injured heart releases microRNAs in EVs, which eventually circulates to the brainstem, modulates neuronal activity, at least sympathetic nerve activity, and is part of the reason oxidative stress and sympathetic nerve activity is tied together. So I'm going to end here, and I want to thank a lot of people that over the years have contributed to, kind of a lifetime of work, and I hope I didn't bore you too much with reminiscing a little bit about some of the older things that we did. Thank you for your attention.
So thank you, Professor Zucker, for this beautiful lecture. So wild. I mean, we started from from the history, and we arrived to the exosomes. So how the physiology and biology change, different approach from observation to mechanism and nanomechanism. Yeah, great. Thank you. So the audience now, you have question? We start from the students. Yes, please. Introduce yourself, and here is the microphone.
Hi, I'm Giovanni Dugo from the third year. I'm a medical student here in the Scuola Sant'Anna. Thank you very much for the lesson. I wanted to ask if these exosomes from the heart to the brain actually do have a role in the evolution of heart failure. Of course they do, but I wanted to know if it is known if they are a positive thing or a negative thing, and if some therapies can be built upon the usage or the stoppage of these exosomes.
Yeah, that's a very good question. Obviously, we don't know all the cargo in the exosomes. They pick up, depending upon how they're made, they pick up many different particles. Some of them even are large enough to carry mitochondria. Now, some of these things may be beneficial and adaptive, and some of them may be deleterious. We do know that if you harvest the exosomes from the venous outflow of injured tissue, you'll find more of them, and you'll find more pro-inflammatory products like cytokines and things like that. But we have not measured everything, and I think they are absolutely a target for therapy, right? Not only a target for therapy, but a delivery system. One of the beautiful things about extracellular vesicles is that you can grow them in culture they're non-immunogenic. You can load them with particles and have them as a delivery system. Eduardo Marban at Cedar Sinai is doing this now a lot. And so they may be a carrier of negative products that make the disease worse, but they're also a way of delivering beneficial tissue, products. So there's a lot yet to be done. In some of the exercise work that we have done, we've shown that just the simple task of contracting muscle enormously increases the outflow of extracellular vesicles. And we know that they have peripheral effects. That's about all I can tell you about that.
Thank you very much.
Okay. Other question from the audience? Even from the physiologists that... No. From the physiology that are here in the audience from the University of Pisa? Yes? Okay. Introduce yourself and thank you.
Hello, everybody. Thank you, Scuola Sant'Anna, for hosting us today. And thank you, Professor Zucker, for your really wonderful lesson today. So my question, I'm Rosario Amato, I'm researcher at the Department of Biology, University of Pisa. My question regards the oxidative stress and synaptic activity. I was wondering how actually radicals can influence membrane potential, if it's something that is direct or indirect effect.
Yeah, it's also a great question. So as you know, the generation of an action potential is mediated by opening and closing of sodium potassium channels. And most of the proteins that that these channels are made of, in the case of potassium channels, Kv 4.3, 4.1, and several of the other isoforms, can be modified by oxidative stress. And there's a whole body of literature showing that you can reduce not only the channel function, but the protein itself by high levels of oxidative stress. There are several individuals at our institution that have shown that, and I think that will modulate not only action potential production, but synaptic release. And I'm sure there are similar mechanisms in synaptosomes themselves. So I think that's the way the thought process is at the present time. Of course, radical stress can alter metabolic activity and certainly the electron transport chain and a whole variety of other things. I mean, we talk about reactive oxygen stress as a bad thing, but we need it. I mean, it's part of normal signaling. If you take it away completely, you're going to have the reverse effect. But in disease states, I think there's high levels of oxidative stress. And when you profile antioxidant enzymes, and we didn't talk about glutathione, which is another major antioxidant, it's very important. But at the neuronal level, I'm sure it's mostly modulation of potassium channel activity.
Okay. Just a really brief second question. What about adrenoceptors? We have some hints about the fact that probably oxidative stress, hypoxia can actually influence levels of adrenergic receptors. So do you think that oxidative stress at the target cell could also influence this loop that goes back and forth?
Yes, I think so. I think that, I mean, so if you have an adrenergic receptor that binds norepinephrine, let's say, there is a component of oxidative stress downstream from that, right? And again, that modulates synaptic activity as well as ion channel activity. And I think high levels of oxidative stress will enhance the effect of adrenergic signaling, in part through modulation of either the adrenergic receptor, you know, beta adrenergic or alpha adrenergic receptor. Yeah.
Thank you so much.
Okay, yes. So now we are open the discussion. No question come. Yeah. Introduce.
Good evening. I'm Sabrina Monturo, and I attended third year of medicine here in Sant'Anna Schools. Thank you for the lecture, really interesting. I would like to ask you if you think that the stellate ganglia can be a target to reduce oxidative stress in that point, and then reduce the oxidative stress and then the sympathetic.
I missed the first part of the question. Could you repeat it?
If you think that sympathetic ganglia, so stellate ganglia, like
Oh, sympathetic ganglia. Yeah.
the stellate,
The stellate.
can be a target to reduce oxidative stress in there. And-
Yes, absolutely, I do. We actually have a grant submitted on that particular issue. I think
You have a good chance to win.
if we get the grant, write to me. No, I think we have shown that NERF2 is reduced in the stellate ganglia of animals with heart failure, and the amount of inflammation in the stellate ganglia is very, very high. So one of the strategies is to reduce that. And of course, this is not just in terms of inotropic state, but arrhythmogenesis as well. And if we overexpress NERF2 and reduce oxidative stress, we can reduce premature ventricular contractions and other arrhythmias. So it's absolutely a very interesting area that we're trying to work on.
Thank you. And the second question, on the first slides, there was the RTX effect on RTX effect on inflammation. I would like to ask if there are any studies that reported its effect on visceral reflexes, like with the barrel reflex functioning or sensitivity.
Yeah, well, so there are studies. RTX was developed as a pain medicine, right? Because it destroys these pain afferents. And it's used clinically now in arthritis and a couple of other sarcomas of the leg and things like that, even in patients with bone cancer. But it's not mainstream therapy, and it depends where you give it. Remember, there has to be TRPV1 receptors expressed, right? So if you look at the barrel reflex, there are not many TRPV1 receptors in the carotid sinus and aortic arch. But on the heart, the spinal afferents, or in afferents from blood vessels that go directly into the spinal cord, it can be used to modulate those reflexes. And we've done some experiments in that case.
Okay, thank you.
Okay. Yes.
Thank you very much, Professor. It was a really fascinating journey, and congratulations for all your achievement. I have a couple of questions for you. The first one is when you show, the idea of focusing on the heart innervation first, instead of all the reflexes that we have altered in the body, was really clever, because I've always wondered what was the first mechanism altering all those reflexes, apart from the loss of cardiac output, and whether it was directly, at least in the myocardial infarction model, the heart damage itself. I was wondering whether do you see any potential pharmacological application far from the heart innervation that is more difficult to be targeted in a real clinical scenario, and whether, because similar results even with survival curves has been obtained also with peripheral reflexes from your colleagues, Schultz, targeting peripheral chemoreceptors, for instance.
Yeah, it's very hard to do systemic things that target either the baroreflex or the chemoreflex. Certainly one can intervene, and as you know, the chemoreflex is sensitized in heart failure, and the baroreflex is desensitized in heart failure. And there's not a lot that can be done. I think part of the issue with the baroreflex is changes in things like sodium-potassium ATPase activity in the neurons themselves, and blood flow in the carotid body, of course. And of course, there's the central changes. And don't get me wrong, I'm not saying oxidative stress is the whole thing. There are many other mediators. But to treat somebody to enhance reflex function systemically is really very difficult. Once there are these changes, you have to treat the heart failure first, and then hopefully that the reflexes normalize, right? It's a little bit easier perhaps in the carotid body because there are specific transmitters that one can target. But you know the carotid body is pretty tiny, and it's very hard to target it. So I think that's still, I mean, this is certainly mechanistic, and I think, but in terms of therapy, it's very hard. Now, of course, one can play around with things like oxygen tension and modulate it from that standpoint, and one could look at things like blood flow. This transcription factor KLF2 is another target that can be used, but so far those studies have not really been done.
And the second question is, apart from chemical denervation, is there, from your experience with this system, a molecular mechanism that we may aim to target with a drug in order not to erase those heart reflexes, but modulate them, in order to have the possibility also to have a reversible effect and also to exploit the physiological function?
We have not found anything that can do that. We can ablate them with RTX. We can, I guess it's possible to modulate them to a certain degree with things like capsaicin, which will desensitize them. Capsaicin is used clinically now for pain. I mean, I think that can all be done, but there's no real target. There's no real way to target those neurons in an intact heart. I mean, lidocaine on the heart, but it's temporary. Yeah.
Okay. Thank you very much. Thanks.
Thank you, Alberto. So, other question from the audience? Everything is clear? I don't think so. And yes, of course.
So, thank you, as my colleague said, for this journey, beautiful journey for 50 years of history in physiology.
Seems like it was just yesterday.
Well, like, am I correct? I mean, where is this nerve 2 coming from? So if I saw correct, if I understood correctly, one of your slides, looks like it is released by the afferent fibers, not by the efferent in the heart. Is that correct? Or I saw afferent.
So we really can't determine that, because when we measure nerve 2, it's in whole tissue, right?
So you don't know
we don't
really.
I mean, clearly in the brain, nerve 2 is expressed in neurons. There's no question about that. Maybe even in glial cells. In the heart, it's in myocytes, and it's very ubiquitous. It's expressed in all cell types.
And can you determine whether it is released by the sympathetic versus the parasympathetic? Do you, I mean, at least that?
No, I think we'd see it both places. Now, the question is it suppressed in one or the other? And we haven't looked at parasympathetic neurons. For instance, intracardiac ganglia, we have not looked at those. But it's certainly in the dorsal root ganglia, in the stellate ganglia, in heart failure, it's depressed. And upregulation can be done.
The local origin cannot be determined yet, right? So
No, but I would guess that every cell type makes it. It's an intrinsic part of the whole biochemical machinery that adjusts these free radicals.
Thank you.
So, I I have a question.
Yeah.
And I worked for a few times with this NRF2 because there are some natural substances like sulforaphane
Yep.
that you have in cabbages.
Yeah.
They activate NRF2, but in a dose-dependent manner.
Mhm.
So, do you think that in the neural stimulation or neural activity, you can have a dose-dependent response, even related to the age of the guy?
You mean in response to an NRF2 activator?
Yes.
Yes, I think you can. You mentioned sulforaphane. We have played with sulforaphane. It's a very potent NRF2 activator. The problem with sulforaphane is that if you extract it from cruciferous vegetables, it's in very small quantities, but it's a very potent NRF2 activator. And an interesting thing about sulforaphane is that it not only dissociates KEAP1 from NRF2, but it also activates the NRF2 gene. So that's great. There are many other. Curcumin is another
Yeah,
one.
curcumin is another one. Yeah.
That can be, and then there are synthetics like bardoxolone methyl and RTA 408 that are now in clinical trials. And I think you can do a dose response to that. There are companies that have put together in pill form all sorts of natural products that have been shown to activate NRF2. But I mean, so far, there has not been a lot of clinical data to show that it's going to increase lifespan or health span. But people are working on exercise training, by the way, we didn't talk about it all, is a really good NRF2 activator.
Yeah. But sometimes when you have a lower activity of NRF2, you can modulate the genes regulated by NRF2 by increasing the acetylation of chromatin.
The circulation of what?
Of histone acetylation. You increase the acetylation of the lysine on the histone. So you relax the chromatin. So if you have a lower amount on NRF2 is active. Actually, we are seeing that if you perform an electrical stimulation, you have an increase of this acetylation of the chromatin. So this could be that the lower doses you can have an activity. That's why I was asking about
it's very possible, and I think one has to be careful about ablating NRF2 completely. You need a low level of oxidative stress of free radicals for normal function, and this is what happened with trying to use NRF2 activators to enhance cancer chemotherapy. Didn't work very well because you need that oxidative stress.
Yes.
But we haven't done a lot of work with dose dependency of NRF2 activators. I think that probably needs to be done a little bit more closely. But there's no question that it's decreased in many disease states. And anything that will increase it and lower oxidative stress should be beneficial.
I have another question. In your opinion, in the regulation of the vagal response, okay, do you think is much more relevant the manganese superoxide dismutase of the cytosolic form?
That's a hard question to answer. I don't know. Certainly, the manganese SOD is very important for dismutating superoxide in mitochondria.
Yeah.
Right. And that's a large source of, especially in the heart, that's a large source of radical stress. But I think not only is cytosolic SOD, the copper zinc isoform, but so is extracellular SOD important. And I don't think anybody has really looked at the balance between all of those that carefully. And in addition to that, of course, we have to be aware that catalase needs to be present as well. So otherwise you'd have high levels of hydrogen peroxide around.
Okay, thank you. So if I think that other questions are not coming, unfortunately, or not, maybe not. However, I would like to thank Professor Irving Zucker. And now Orizzonti is not over, because now we have something that come from our delegates, students. You have a ceremony that involve all that each speaker receive this certificate from the school, signed by the rector. Where is the? So I leave the word to the student.
One of us. Thank you. Thank you very much. I'll probably display this in my office. Oh, very nice. Thank you. I will write it here, but I guarantee you, you won't be able to read it. And you have a pen? Let's see.
E io come fisiologo qui della Sant'Anna devo fare anche il saluto alla comunità dei fisiologi pisani. Lunga vita alla fisiologia, questa scienza che sembra essere andata fuori moda, ma che ha sempre tantissimo da dire. Anni fa no, assistemmo quasi alla sua scomparsa e poi finalmente si è capito che non se ne poteva fare a meno.
Now we have
Okay. Okay, great.
and we will put Mr. Becker again to be there.
Thank you.
Mr. Becker. So this will be very